Organ transplantation, immunology, tissue typing, and human measure histocompatibility complex. Histocompatibility testing, the rejection process. Tissue typing. All somatic cells have a surface glycoprotein acting as a self-marker for these cells. These surface markers were originally detected on the leukocytes and were named human leukocytic antigens, now universally referred to as HLA antigens. Mixing the tissues of a prospective donor and of the recipient and tested for compatibility prior to transplantation. The self markers are responsible for triggering the immune reaction leading to allograft rejection. Tissue typing techniques. Mixed leukocyte reaction. Culturing lymphocytes from the donor together with those from the recipient. Microcytotoxicity assay. Utilizing serum with known anti-HLA antibodies that recognize particular HLA loci, HLA-A, HLA-B, HLA-C, HLA-DP, HLA-Q, DQ, HLA-DR in order to match genetically similar individuals for tissue transplantation. The goals of the histocompatibility testing. Immediate goal, serological cross-matching between the recipient and the potential donor to prevent hyperacute rejection using the serum from this and that. Long-term goal, HLA matching by minimizing the genetic disparity between the recipient and the donor to maximize the length of the graft survival. The major histocompatibility complex, MCC. The HLA antigens are genetically controlled by loci on the short arm of the chromosome number 6. This area on the chromosome number 6 is termed as the major histocompatibility complex. It is a set of cell surface molecules encoded by a large gene family which controls the major part of the immune system in all vertebrates. The major histocompatibility complex functions. The major function of the MHCs are to bind to peptide fragments derived from the pathogens and display them on the cell surface for recognition by the appropriate T cells. MHC molecules mediate interactions of leukocytes, which are immune cells, with other leukocytes or with body cells. The MHC determines compatibility of donors for organ transplant as well as one's susceptibility to an autoimmune disease via cross-reacting immunization. The major histocompatibility complex divisions and functions. The MHC gene family is divided into three subgroups, class 1, class 2, and class 3. The important antigens in transplantation are the class 1 products, HLA, B, and C antigens. Foreign class 1 and class 2 antigens stimulate the recipient immune mechanism and trigger lymphocyte sensitization, starting rejection of the transplanted organ. The rejection process, cellular mechanism and humoral mechanism. The cellular rejection mechanism. Cellular and soluble graft antigens will affect the recipient's immune system by leading to the recipient's immune system will attack the antigen by macrophages and dendritic cells and also will activate the and, and proliferation of the T helper cells. Both of the attack and the proliferation will produce the interleukin 2. That interleukin 2 will lead to activation and proliferation of the cytotoxic T lymphocytes. This activation of the T lymphocytes, cytotoxic T lymphocytes, will lead to direct attack of the transplant tissue and in turn damage to the cells carrying the foreign HLA antigens. The humoral rejection mechanism. Presence of complement fixing cytotoxic antibodies prior to transplantation leads to hyperacute graft rejection. Attacking the graft's vascular endothelium leads to platelet aggregation and thrombosis. Types of graft rejection. Hyperacute graft rejection, the onset is immediate. The mediator is the humoral antibody B antibody. The gross graft appearance, it is flaccid and cyanotic. The treatment is removal. The prognosis graft loss. The accelerated type of rejection. The onset 2 to 5 days. The mediator humoral AB. The gross graft appearance flaccid and pale. The treatment is by removal. Prognosis graft loss. The acute rejection occurs 7 to 30 days. The mediator cellular and humoral, both of them. Gross graft appearance, graft edema, cortical necrosis, maybe graft rupture. The treatment, corticosteroids, OKT3. The prognosis is good. Chronic rejection, the onset is variable. The mediator is cellular. The gross graft appearance, it is shrunken and fibrotic graft. Treatment, no way to treat. The prognosis is bad. OKT3 in treatment of acute rejection. OKT3 is a brand name for muromonab CD3. It acts by blocking the function of the T cells. It reacts with and blocks the function of a molecule called CD3 in the membrane of the T cells. The binding of OKT3 to T lymphocyte results in their early activation, which leads to cytokine release followed by blocking T cell function. After OKT3 therapy is over, T cell function usually returns to normal within a week. Good luck and best wishes.